Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. Let me bring up the syllabus. And let me state that I also thought about the changing the final date. And I think it's too late to change the final because this class is online and some people won't, well, some people probably won't even ever see this recording, but some people certainly won't see it until the weekend. So I really don't want to change the final date. So I've decided to leave it as it is scheduled in the syllabus. Any questions about that? All right, let's go to the syllabus. Where am I? So the schedule is for the final to be finished by 11:59 p.m. Wednesday, June 19th. On this final, there will be heavy coverage. About 50% of the material will be from the material after the last quiz, meaning it will cover chapter 7, chapter 13, chapter 14, and lab module 9. Now, 50% of the other questions will come from the material that you have been quizzed on. And that will be labs 1 through 8, and then chapters 1 through about 8. Any question about any of that?
I have a question, Professor.
Uh-huh.
Will there be any essay questions on the final exam?
There may be about 2 essay questions.
Okay, thank
They
you.
could be just short answer, but there's at least 2 questions, I think, which will be either you have to fill in the blank or it'll be write a couple of sentences. I don't have anything longer than a small paragraph, but and that will be in the final A. Final B will be multiple choice, where you will be given a question and then you select the answer. Any question about that? Most of the questions will be final B, which will be multiple choice. But I believe there are 2 questions that are either fill in the blank or essay.
How many numbers would be like the A, quiz A, and
There's
quiz?
not more than 4 questions in quiz A.
Okay. How about the
Okay. But some of them, it might show you a table and then give you some test results. And then you have to identify the species determined by the table. And some of them might ask you a question and then you have to fill in a blank or write an essay, something like that. Okay. Final B will be most of the questions and it will be multiple choice. All right. Let me also state that your unknown notes pages is going to close on Friday, 11:59 p.m. If you ask a question on Friday, it has to be a 1-day test because 2 days you will not have enough time to incubate it or anything longer than 1 day. I can get you the results on Saturday. Your unknown project report has to be due by 11:59 p.m. Saturday, June 15th. And you can turn it in early. This project will be checked for, what do you call it? Plagiarism. Any questions about the unknown work? Let me remind you, if you haven't put up the last results that I sent to you in your unknown notes page and then tidied it up, that the unknown notes page will be graded for 5 points. So make sure you get your last results up. I think that's it. So let's go ahead. What I'm going to be doing today is going over the material. That will be a major review for the final. It won't be the only review, but a lot of the questions will be reviewed. Not all of them, but a lot of them. And if you want to, let's see if I can share my screen here. Excuse me. If you want to study, the best thing to do for studying on the final is to go over the objectives. Let's go down to chapter 1. Well, maybe lecture 2 because I went past it. And take a look at the objectives. Now, on some of the chapters, I've only got it in the study guide. The objectives will be the start of the study guide. And go over the objectives. If you know all of the objectives, then you should do very well on the final. I believe all of the questions are covered in the objectives. Any question about any of that? And then, of course, go over this review, and then go over your notes. If you have more time, you can then go over the textbook reading. Any question about that? There will be one question from each chapter. Let's see if I can show you that. There's one question from each chapter in the earlier chapters. So one question from chapter 2, one question from chapter 3, etc., all the way down to chapter 8. Chapter 8 was a very large chapter, and we took 3 days to cover it. So there will probably be a few more questions, 2 or 3 questions from chapter 8. And then from chapter 1 introduces a whole bunch of concepts that we later discuss in the class. And then chapter 1 also goes over the history of microbiology. And I'm very fond of the history of microbiology. So I tend to ask a few more questions on chapter 1 because of what it's covering and how it's covering it. Meaning that if I ask a question about Koch's postulate, it was introduced to you in chapter 1. We did talk about Koch's postulate and a few other topics 3 times throughout this term, but they were all introduced in chapter 1. So there will be more questions from chapter 1. For the history of microbiology, all you need to know is the major historical events and then who discovered them. You do not need to know the year, with the exception of the very first item we discussed in the history of microbiology, and that is you should know about how long ago the first bacterial fossil records are on the planet Earth. Okay, that's the only year you need to know. All the other years are just brought in there for, I don't know, helping me organize it. Any question about any of that? All right, so from the chapters that you've not been quizzed on, that'll be chapter 14, 13, chapter 7. There will be multiple questions, numerous questions from each chapter. I I don't know, something like at least 3 and potentially 5 questions from each chapter. And then lab 9 will have a few more questions as well because we weren't quizzed on lab 9. Oh, and then your unknown project. You'll have a few questions on your unknown project too. Any question about any of that? There will be no questions from the infectious disease project because you each had different diseases. All right. If there's no question, I'm just going to go from the back, reading chapter 7, and work forward. I suppose I should take a look at lab 9 before we begin. Well, in lab 9, you should know the Einvik reactions, what the indole test, methyl red test, and Voges Berskira test, what they are when they are positive. And it's pretty easy to remember that because all of them are red when they're positive. And when they're not red, they're negative. I believe all of these are yellow when they're negative. In the citrate test, it is blue, and you'll see growth on the tube when it is positive. And when it's negative, there shouldn't be any growth, and it's going to be green in color or unchanged. And you can also say indole, methyl red, and Voges Berskira. In their negative, they're unchanged. You should know about carbohydrate fermentation test, growing the bacteria in a PR carbohydrate tube, which could be PR glucose. And if it's positive, it'll be yellow. There may or there may not be also gas production. If it's yellow, we abbreviate it with A for acid production. And then if there is gas, it's AG for acid and gas. You should know about the motility test. If the bacteria is only growing in the stab, then the bacteria is not motile. If the bacteria flares, the growth flares out of the stab, meaning it looks something like, let's see if I can get paint here. If it flares out away from the stab, then that's slightly motile. And it can also be cloudy everywhere, in which case it's very motile and the cells have gone everywhere in the tube. Any question about that? Hydrogen sulfide production, the tube is black if it is positive, and it is not black, generally unchanged or yellowish if it is negative. You should understand about the TSI, triple sugar iron test. And this is a very difficult test because so many things are going on. It's looking at a number of different test results, looking at glucose fermentation, and then either or both sucrose and lactose fermentation, looking at the production of hydrogen sulfide, and looking at the production of gas. If there's gas production, there will be cracking or a bubble or the lifting of the auger in the tube. And if hydrogen sulfide is produced, it'll turn black. If only glucose is fermented, then the slant will be pink and the butt will be yellow. And if lactose and or sucrose are fermented as well as glucose, then it'll be yellow in both the butt and the slant. You should know the SIMS test looks at 3 different tests: sulfur, production, indole production, and then motility. If there's sulfur production, the tube will turn black, especially in the bottom. Indole, it'll be red at the top. And if it's motile, you'll see the bacteria growing out of the slant. Growth on McConkey's agar will be showing that it's a gram-negative. It can grow on McConkey's, and it'll turn the agar as well as the colonies pinkish color if the bacteria can ferment the sugar lactose on hectone enteric agar. If it can ferment if it grows on the plate, it's gram-negative. And if it can ferment the sugar lactose, it'll be a salmon color. If it is a bacteria that can produce hydrogen sulfide, the colonies will appear black. And then use biochemical analysis to solve case studies. And in this case, it doesn't need to be a gram-negative bacterial infection, but if they ask a question about lab 9, it will be a gram-negative bacterial infection. Any question about that? So in this lab, we're mainly looking at two families, the family Enterobacteriaceae and the family Pseudomonaceae. They're both gram-negative, non-spore-forming bacilli, but there are some differences. The family Enterobacteriaceae, all the members can ferment the sugar glucose. The Pseudomonaceae, all the members do not ferment the sugar glucose. The other difference is Enterobacteriaceae, all the members of the family are oxidase negative, and the Pseudomonaceae family, all the members are oxidase positive. Any question about any of that? All right, that's it for lab 9. Let's move on to chapter 7. So you should always know the terms for the lesson. You should know that bacterial cells die at a constant logarithmic rate. So if you plot it, semi-log plot the number of surviving cells over the time, the cells will die at a constant logarithmic rate. There are a number of factors that affect the microbial death. The number of microbes present at the beginning, the more microbe present on the beginning, the longer the treatment has to be. There are environmental influences that can affect it. Name the organic matter. The organic matter can inactivate the chemical agent like bleach. The temperature of the treatment can affect the microbial death, and then the presence of biofilms. Generally, higher temperature is better to kill, and then the absence of a biofilm is better to kill. I've already mentioned a little bit about time. You need sufficient time to kill all cells, regardless of what the treatment is. And then the microbial characteristics can affect the microbial death rate as well. Like the bacteria, Mycobacterium, has mycolic acid in its cell wall. And so the mycolic acid can prevent water-soluble disinfectants from getting at the cell and then easily killing them. Any question about any of that? All right, know the terms. You should know the difference between a biocide, germicide, bactericide, fungicide, virucide, and then a bacteriostatic agent. Biocide will kill the cells. Bacteriostatic agent will only inhibit the growth of the microbe. Once you remove the biostatic agent, growth may continue. You know about sepsis and asepsis. You should know that antimicrobial agents kill cells by generally 3 activities. 1, alter the membrane, damage the membrane. 2, damage intracellular proteins. And then 3, damage nucleic acid. You should know about the physical methods for controlling microbial growth. You should know that heat kills microbes by denaturing proteins, although if you use high enough heat, you can actually char the cells, such as flaming and incineration. You actually char the cells. You should also know that wet heat involves boiling and autoclave and steaming. This is more efficient at killing cells than baking. And we all know that when you put your hand through steam, you'll get burnt immediately. And you can put your hands in a hot oven and pull out what you're cooking, and you don't burn yourself. Actually, once when I was teaching middle school students, I took them on a field trip, and the field trip guide said, "Would you like to walk through the oven?" This had this huge oven, and almost all the students went walking through the oven. And I just said, "No, I'm going to walk around." So the point is, you can actually walk through a hot oven and not get burned. How we reliably kill things in a microbiology lab is we autoclave things, that is steam under pressure, generally 15 pounds per square inch of pressure at 121 degrees Celsius. Will kill all microbes, at least in 1 liter, including endospores, after 15 minutes. This isn't talking about prions, but all cellular microbes will be killed after about 15 minutes, at least in 1 liter size. Any question about any of that? You should know about pasteurization and the 2 ways of doing it, and know that ultra high temperature milk is not pasteurized milk. It's actually sterilized milk. Pasteurized milk does have microbes in it. It just does not have any microbes that are human pathogens or microbes that can spoil the milk, at least right away. But it does have microbes in it, and so the pasteurized milk will have a shelf life. Dry heat, baking, as the cat is showing you, doesn't kill you immediately, doesn't burn the cat immediately either. Filtration, another way of controlling microbes by passing through things through a filter, but block the cells. Low temperature keeps microbes from growing, but it may not kill, certainly doesn't kill all microbes. And then fast freezing doesn't tend, in liquid nitrogen, doesn't tend to kill microbes. High pressure does kill vegetative cells, not endospores. And we use high pressure when we want to preserve the color, the flavor, and the nutrient value. So some juices are preserved by high pressure. Desiccation, an example of that would be like dried fruit, dried vegetable. High osmotic pressure, which is similar to desiccation, where you have high salt, high sugar content, like molasses. The bacteria falls in the molasses, the water will be pulled out, and that will preserve the molasses, the food, causing plasmolysis. That would not kill endospores, but it would kill certainly many bacteria like E. coli cells. And then it would prevent all bacteria from growing. radiation, another physical method for controlling microbial growth, ionizing radiation, X-rays, gamma rays, electron beams have a lot of energy. And when that energy is absorbed by an atom, that will, it'll be absorbed by the electron. That'll give the electron enough energy to leave the atom. And that will cause an ion, why it's called ionizing radiation. Those ions can then bump into other molecules, damaging the molecules, damaging the cell, especially damaging nucleic acid. UV light is non-ionizing radiation, which can also damage DNA, creating thymine dimers. You should know the terms. You should know about chemical methods for controlling microbes. Disinfection, the destruction of vegetative pathogens on an object. Some different types of disinfectants. An antiseptic is when you use a chemical on living tissue. So if you use bleach on an object, we call it a disinfectant. If you use bleach on living tissue, which isn't done very often, but it used to be at least one of the good early treatments for necrotizing fasciitis, we call it an antiseptic. Comparing the effectiveness of various antiseptics, soap and water does not kill very many cells. It is a very weak disinfectant or antiseptic. And then tincture of iodine with about 90 seconds exposure, you reduce the population of cells down to 10%. You know the microbial characteristics, creating how sensitive the cells are to the antimicrobial agent, with viruses with lipid envelopes and gram-positive bacteria being the two most sensitive, prions and endospores being the most resistant. Now, it depends on the specific bacteria, what characteristics it has. The mycobacteria are resistant to many chemical disinfectants because of the mycolic acid in their cell wall, but the mycobacteria are sensitive to alcohol, which is a very weak disinfectant. Generally, we call it a degerming agent, not a disinfectant or an antiseptic. And the mycobacteria are actually very sensitive to alcohol because the alcohol can get right through the mycolic acid. Any question about microbial characteristics and the hierarchy of resistance of the major microbial groups to a chemical biocide? All right, that's it for chapter 7. Let's move on to chapter 13. Viruses, viroids, and prions. That might not be it. Looks good. Yeah, that's it, I think. Nope, it's this one, I think. All right. So know the terms of the chapter, understand the characteristics, structure, and the classification of viruses, understand viral multiplication, meaning viruses have a life cycle, and it depends on the species, how many life cycles they have, not the species, the kingdom of virus. Like the bacterial viruses have 2 life cycles. And then understand about prions and viroids. The goal of the virus is simply to replicate. How they do that is they attach to their host cell invade it, and then take over the host cell and use it to replicate. Any question about any of that? You should know that viruses are inert when they're not within their host cell, meaning they have little or no metabolic activity. Viruses always have a nucleic acid molecule for their genome, which can be either DNA or RNA, but never both. And they always have a protein coat surrounding that called the capsid. And how they replicate is they use host enzymes, nucleic acid, amino acids, ATP to replicate. A virus may also have a lipid bilayer, what we call an envelope, around the protein coat. It depends on the family virus, whether it does or does not. We call it an envelope virus if it has an envelope, a naked virus if it does not have an envelope. The host range is the range of cells that the virus can infect. This is determined by the specific host attachment molecules that we call ligands or adhesions, which bind to specific host cellular molecules that we call receptors. Generally speaking, most viruses only bind to cells of one or a few species. There are some exceptions, and that's because of the specific binding of the virus to the host cell. The range is the range of cells in species that the virus can infect. Bird flu would be an example of a virus which is not narrow. It can infect many different birds, maybe all birds, I don't know, but certainly many different birds and many different mammals. Any question about any of that? I don't know if it infects any other animals, but it may only infect warm-blooded animals. You should know about the relative size of virus compared to a prokaryote cell. You should know about virus structure and that they can have DNA or RNA, and this DNA or RNA can be double-stranded or single-stranded. And if it's single-stranded RNA, you have to state whether it's positive single-strand RNA or negative single-strand RNA. The positive single-strand RNA genome can be read by the ribosome. The negative single-strand RNA genome cannot be read by the ribosome. Any question about any of that? Close that. You might get some noise from outside. I talked about that. You should know the viruses come in 3 different shapes: helical viruses, polyhedral viruses, and then complex virus, where in a complex virus, the virus can take on essentially any shape. Now, most viruses have one shape for the species, but different species can be different shapes. You should know that T4 virus is the virus that has been studied more than any other virus. And here we're looking at T4. Let me mention briefly about that. Some viruses can change or mutate frequently, when different viral strains can therefore infect the same individual more than once. An example being influenza virus, another example being COVID-19. I've heard that there's been documented cases of people getting COVID-19 5 different times, which, by the way, you don't want. The more times you get COVID-19, the more likely you are to get what is called the complex long-term COVID. I forget what they call that, but long-term COVID. In viral taxonomy, we generally group viruses into families, into genus, into species, and subspecies. We also group them into the kingdom that they infect. We do not tend to group them into other groups because we just don't know enough about viruses. Their genome isn't big enough and they evolve very quickly. And so we just don't know the evolutionary history of viruses to group them into like phyla. Any question about that? The virus family name ends in viridae. The genus name ends in virus. The species will be the common species name, English species name, by the way. And then the subspecies will be designated by a number. The subspecies is really important. Like with poliovirus type 1, it causes the disease polio. Poliovirus type 2, we use it as the live vaccine to prevent getting the polio disease. An example of a virus family is the herpes viridae. An example of a genus will be herpes simplex virus. And then you have the subtype herpes simplex virus type 1. Any question about that? All right, bacteriophages have two life cycles. The lytic life cycle, where the virus replicates inside the host cell and will lyse the host cell to release the newly replicated viruses. The other life cycle we have is a lysogenic life cycle, where as long as the virus remains in the lysogenic life cycle, the cell will remain alive. The lytic life cycle goes through 5 stages: the attachment of the virus to the host cell, penetration, biosynthesis, maturation, assembly of the phage particles, and then release, where the virus actually releases the enzyme lysozyme, which will then digest the cell wall. Water will move into the cell, which will eventually burst the cell, releasing the newly replicated viruses. You know about burst time, the time from initial infection to the time the cell burst, and then burst size, the number of newly replicated virions. That will be dependent on the species. The burst time will be dependent on the species and the temperature you're growing the viruses at. Lysogenic viruses, when they infect a bacterial cell, the virus has to decide immediately what to do. It can enter the lytic life cycle, in which case it'll go through the normal lytic life cycle, or it can enter the lysogenic life cycle. If it enters the lysogenic life cycle, the genome of the virus will integrate into the host cell's chromosome, in which case we call it a prophage. And as long as the lysogenic life cycle continues, the host cell and the virus will be living in symbiosis, meaning host cell will remain alive. How that happens is there's a suppressor gene of the lysogenic life cycle, and the suppressor gene shuts down the lytic life cycle. And the lysogenic life cycle, the virus only replicates when the cell replicates. And then the daughter cell, each cell will get the prophage because it's replicated. The prophage is inside the host cell's chromosome. For reasons that are not all fully understood, after even more than 100 years studying it, the lysogenic life cycle can end and the prophage is excised from the cell, and then the lytic life cycle can begin. Any questions about any of that? All right, some consequences of lysogeny. If we have a bacteria cell, which has a lysogenic virus in it, that bacteria cell will be immune to infection by the same species of bacteriophage, meaning other bacteriophage cannot infect that cell. And then the other consequence of lysogeny is this cell picks up new DNA, the genome of this virus, and that can change the phenotype of this cell once the genome of the virus is expressed. The best known example is the bacteria Corynebacterium diphtherium. This bacteria only produces the diphtherium toxin when Corynebacteria diphtheria has a lysogenic virus. The diphtherium toxin is actually a gene from the lysogenic virus. Any question about any of that? All right, let's move on to animal viruses. They do follow multiplication, very similar to bacteria viruses, with a few differences. The main difference is well, there's two, I guess, main differences, and that is they can have different life cycles. The two major are the budding life cycle and the lytic life cycle. The lytic would be the same as a bacterial lytic life cycle, but the budding life cycle is totally new for an animal virus. There are some other life cycles. We'll briefly mention the latent life cycle and the persistent infection life cycle. But the two main are budding life cycle and the lytic life cycle. The other difference is where the virus can replicate. In a eukaryote cell or an animal virus, it can replicate in the cytoplasm or it can replicate in the nucleus. Of course, a bacteriophage can only replicate in the cytoplasm because bacteria do not have a nucleus. Attachment of the cell will be similar, but the penetration of the virus will be different. And with animal viruses, they can gain entry into the cell either by endocytosis or by fusion of the envelope of the virus with the cell membrane. So only envelope viruses can engage in fusion, but the envelope can fuse with the cell wall, and then let's make it this one right here, the cell wall, instead of going across like that, will then be part of the envelope of the virus and then come around. And that will allow the viral genome access to the cytoplasm. All right, talked about that. You should know that there are 7 families of DNA viruses that can cause human disease. You do not need to know or memorize this table. For DNA viruses, the rule of thumb is they replicate in the nucleus. I don't know of any exceptions. And that's because in the nucleus of a eukaryote, we have everything that the virus needs to replicate. The replicate virus will then move into the cytoplasm. Messenger RNA, viral messenger RNA will be transcribed and move into the cytoplasm. The capsid proteins will be made, and then the capsid proteins need to get together with the replicated newly replicated DNA strands of the virus to make the new progeny virions. This slide is showing you multiplication of a DNA virus in greater detail. Let's move on to the RNA family of viruses. I believe there's 15. 15 families of RNA viruses which cause human disease. Once again, you do not need to memorize this table. You should know a few of the different families of viruses and then the diseases they cause. I don't know if I mentioned it, but Picornaviridae is the family of virus that is the #1 cause of human colds. So you should know that. And that there are 3 families of viruses that cause human colds. Picornaviridae is just the most common one. Adenoviridae is another. And then the third is a DNA virus, which is the adenoviridae. Coronaviridae is the second. Polio is a Picornaviridae, as well as hepatitis A. And then many of the viruses that cause human colds, but not all, come from family Picornaviridae. You should know a little bit about Coronaviridae because we talked about it. I'll go over that in a little bit. Orthomyxoviridae, you should know, causes the human flu. Oh, and then Retroviridae, you should know about that family too. The RNA family of viruses, as a rule of thumb, replicate in the cytoplasm. There is one exception, which you do need to know. Family Retroviridae is an RNA family of virus, but it does not replicate in the cytoplasm. It actually replicates in the nucleus. And that's because it has a DNA intermediate. Any question about any of that? All right, you should know the negative strand RNA virus genome cannot be read by the ribosomes to be translated into protein. Instead, the negative strand RNA makes a copy of itself, which would be the positive strand RNA. And the positive strand RNA can be used as the viral messenger RNA to translate the messenger RNA into protein. Any question about any of that? So this slide is showing you how the different viruses can be replicated. And you should know how a positive single-strand RNA virus replicates, as well as how a negative single-strand RNA virus replicates. All right, let's briefly talk about family Retroviridae. It causes HIV-1 and HIV-2 cause AIDS, and then feline leukemia viruses causes leukemia, where the cat leukemia gets so bad that it weakens the cat's immune system, and then the cat can get what's called kitty AIDS. Family Retroviridae is unique because it has the enzyme reverse transcriptase, which is an RNA-dependent DNA polymerase where the reverse transcriptase can read the positive single-strand RNA genome to make, in 2 steps, double-stranded DNA, which this will then integrate into the host cell's chromosome and become a permanent part of that cell's DNA. So once a cell becomes infected with the retrovirus, it will be there for as long as that cell is alive. I didn't mention it, but I can tell you that the human genome has 5 or 6 retroviruses in it, but fortunately, all of these retroviruses are mutated and then non-functional. In the mouse genome, they do have a family of viruses, which actually is a carcinogenic agent. This virus, which is integrated into the mouse genome, tends to cause breast cancer in older female mice. Doesn't hit the younger female mice because you do want the DNA of the cells to replicate. All right, any question about any of that? The reason why the RNA family of viruses replicates in the nucleus is because it has a DNA intermediate, which will then integrate into a host cell's chromosome. All right, let's briefly talk about coronaviruses. They were well known to humans long before COVID-19 made its appearance in 2019 because they can cause human colds. And they also cause some other serious diseases like SARS and MERS. SARS first appeared in 2003, and fortunately, the medical community got control of it, put the people who were infected under quarantine, and then SARS never spread further. And at the moment, it's currently extinct in the human population, but it could every once in a while come back, causing SARS disease, where about 10% of the patients die. MERS is worse. MERS stands for Middle East Respiratory Syndrome. About 36% of the patients who get MERS tend to die. Fortunately, MERS does not spread patient to patient. And whoever gets it is usually someone who's in close contact with camels. So COVID-19 appears to be a virus which recombined 2 bat coronaviruses and then recombined with a pangolin coronavirus to make COVID-19. I think we'll skip all of that and just move on to viruses and cancer. 10 to 15% of human cancers tend to be caused by viruses. Viruses cancer, what it is just unregulated growth of cells. Viruses can cause cancer in 2 ways. First, if the virus integrates into the host chromosome, causing a provirus, that can cause a permanent genetic change in the cell, turning on genes that should not be turned on, turning off genes, or even mutating genes that should be functioning. And this mucks up the DNA, causing mutations to perhaps happen or start. And then if you get enough mutations, 5 or 6 of them, you can have the cell become cancerous. Any question about any of that? And then the second way that viruses can cause cancer is if the virus brings to the cell an oncogene. An oncogene is a normal cellular gene involved in cellular growth. This is a mutated normal cellular gene involved in growth, and the mutation causes the cells to grow uncontrollably, causing the cancer. Any question about that? All right. Another life cycle you should know for animal viruses is an animal virus can enter the latent life cycle To my knowledge, only the budding viruses ever entered the latent life cycle. I don't know if that's entirely true or not, but at least most, if not all viruses, if they can enter the latent life cycle, they can also enter the budding life cycle. The budding life cycle is where the virus is actively replicating at low numbers so that the virus does not kill the host cell, and then the virus can then bud out of the host cell later. But the point is that while the virus is replicating, it's the budding life cycle. The virus can stop replicating, and depending on the species, can leave the budding life cycle and then enter the latent life cycle, where the virus is essentially just sitting there doing nothing, not causing disease, not replicating. A latent virus can obviously lose the latency, meaning leave the latent life cycle and then re-enter the budding life cycle. An example of that would be shingles. When the patient first becomes infected with the virus, they'll get chickenpox. The virus will be replicating while the child has chickenpox, and it usually hits children, although it doesn't have to. And then in about 20%, 25% of patients, the virus can leave the latent life cycle and then re-enter the budding life cycle, where the virus is replicating. But in this case, the virus only replicates in nerve cells or central nervous system cells. Oh, neurons, that's the term where I'm trying to think of. Virus only replicates in neurons, and when the virus starts replicating again, we call it shingles. Whenever the virus replicates more than the first time, we will call it shingles. The trigger is some disease manifestation caused by a stress, like when an individual gets cold sore. The stress of the cold can cause the virus to leave latency and then enter the budding life cycle, in which case you'll get a cold sore. And then after the cold sore disappears, the virus goes back into the latent life cycle. Any question about any of that? The persistent viral infection is a virus, an animal virus, that's similar to the budding virus, in that the virus is generally made at low numbers, and then the virus is packaged inside an envelope or a membrane-bound organelle called a vacuole. And the vacuole can move up to the cell membrane and then fuse with the cell membrane, dumping its contents outside the cell. And that would be a persistent viral infection. In the old days, AIDS caused by HIV could be a persistent viral infection where the viral number increases over time. Initially, it's low, but it increases over time. And then the patient's immune system goes away, causing the signs and symptoms of AIDS. Like I said, this is the older days. Now we have medication to keep the patient alive. Back in the older days, like 1980, when the disease first appeared in the United States, the patient would eventually develop AIDS, and then once they got AIDS, they would die. Nowadays, multiplication can keep an AIDS patient or an HIV positive patient alive for most of their lifespan. Not entirely a true human lifespan, so it's a little shorter. Some persistent viral infections are tolerated, like cytomegalovirus. About 50% of Americans came down with a cytomegalovirus infection, and in some of these patients, the virus will enter the persistent life cycle, will always be shed at generally low numbers, and the patient tolerates it because it's not doing that much damage. Any question about any of that? All right, this is showing you the persistent viral infection, an acute viral infection, and then a latent viral infection. An acute viral infection is where a patient gets exposed to a virus and then quickly, relatively in a few days, develops signs and symptoms as the viral load increases to a high number. What's good about the acute viral infection is the viral number quickly decreases, and then the time that the patient has the viral disease is relatively short. So the acute viral infection, the number of newly infected virions up here, and simply the time on the x-axis. That differs from a latent infection where you initially have an acute infection where the child gets chickenpox, which happens fairly quickly after the child is first exposed. And then the virus load gets very high very quickly, but then it doesn't last long, and very quickly the viral load starts to decrease. Then the virus enters the latent life cycle, and there is no disease, no replicating virions. And then the virus comes out of latency, re-entering the budding life cycle. And usually the second and later events, we will have a less steep curve because we're producing less viruses than the acute viral infection, such as when a patient is in shingles. They first had chickenpox, and then they went into latency. When the virus came out of latency, it re-entered the budding life cycle, causing shingles. A persistent viral infection is a virus that starts at low numbers, and over time, the number of microbes increases. The virus tends to last a long time or persistently, meaning long time or essentially forever. Most of them, it's for a long time, like mononucleosis. The patient may be sick for several months, but eventually it is thrown off. Some persistent viral infections can be tolerated. Like I said, some cytomegalovirus infection. The patient is always budding cytomegalovirus at low numbers. In that case, the virus tends to plateau at a given level, meaning no further number of virions, because if it were to get more and more, you would eventually get signs of disease. And we remember we're talking about asymptomatic disease, where the patient has the microbe, can spread the microbe, but the microbe is at low enough numbers in the patient that it does not cause any damage to the patient. No damage, that means there's no signs or symptoms. Any question about any of that? All right, I already talked about that. All right, let's talk briefly about prions. They are an infectious protein or a proteinaceous particle. They can be inherited, transmitted by ingestion, transplant, like an organ transplant, and surgical instruments. Prions tend to cause spongiform encephalopathy, where there will be vacuoles developing in the brain of humans and animals. We call it spongiform encephalopathy in animals, although scabies is a disease that's called scabies, which is the same thing, but we don't call it spongiform encephalopathy. In humans, we don't tend to call it spongiform encephalopathy. We call it either the Creutzfeldt-Jakob disease or Gerstmann-Straussler-Schindler syndrome. fatal familial insomnia, or Kuru. Kuru was a disease found in the Four tribe, and it was because they had the habit of when somebody died, the relatives would cook the body and then eat the body. And you notice when I mentioned the body is cooked, so that will kill most microbes, but it does not kill prions. The prions will still be there and can infect a patient, causing essentially leading to death. The mechanism of how a prion replicates is the prion form of the protein comes up to the normal form of the protein, which is a protein needed by the nerve cell. And then the prion causes the normal protein to change its folding pattern into the folding pattern of the prion protein. The prion protein then accumulates in the cell. As more normal protein is made, it'll make more prion protein. And that will cause plaques to form in the brain, eventually leading to cell death, meaning the nerve cells will start dying. All right. Plant viruses are similar to bacteria viruses in many ways, other than that plant viruses tend to infect a plant cell by a wound in the plant or by a parasite such as a nematode, a fungus, or an insect, damaging the plant, allowing the plant virus to get into the cell. Infected plant viruses do not cause disease in humans, but only in plants, including economically important plants. And then the virus can spread either from, let's say, an insect bites the infected corn, the insect picks up the plant virus and then bites another corn, causing the virus to move that way. And then viruses can also spread by seeds as well as pollen. You should know that viroids are a naked RNA molecule, somewhat similar to an RNA virus, but there's no capsid around this RNA. And it's an infectious RNA molecule that can infect a cell, causing plant disease like potato spindle tuber disease. Fortunately, there are no viroids known in animals. So there's no viroid that cause diseases in animals. Any question about chapter 13? Let's move on to chapter 14. In this chapter, you should know the terms. If you know the terms, you'll learn 50% of this chapter. You should also understand what a normal microbiota is and its relationship with its host. You should understand the stages of disease development and understand about nosocomial infections and how to control them. You should know the terms. There are large communities of microbes found in association with all animals and plants, and these are called the species normal microbiota. Let me iterate that human normal microbiota is slightly different, or at least different from cat normal microbiota. Like cats have a Bartonella henselae, part of their normal microbiota, and humans do not have that as part of their normal microbiota. And if they get Bartonella and henselae, then they are likely to get cat scratch fever, that disease. So actually, this species, which is important, normal microbiota in cats, is actually a human pathogen. When the normal microbiota is its balance with the host is disrupted, that can cause to signs of disease, such as when you take antibiotics, it disrupts the normal microbiota, and that can give the patient either constipation or diarrhea. Fortunately, the normal microbiota reestablishes, and then the diarrhea or constipation usually goes away within 2 or a few days. The human normal microbiota is found on all external surfaces of the skin, inside the mucosal membranes like the eyes, the nose, the mouth, the gastrointestinal tract, the lower reproductive and urinary tract, meaning the lower urethra has a normal microbiota. The normal microbiota differs from the transient microbiota in that the transient microbiota can last for maybe a few hours, days, or weeks, but it will eventually go away. And why it's called the transient microbiota. I gave the example that I once had Staphylococcus aureus on my skin, and fortunately, it went away so that I lost Staphylococcus aureus as part of my skin. Now, about 20% of Americans have Staphylococcus aureus as part of their normal microbiota. And these 20% of Americans are much more likely to get a skin boil, the skin boil being caused by Staphylococcus aureus. The normal microbiota is not usually found internal, like internal organs, inside the blood, inside the heart, or inside the bone. And if it is, it's usually because of an infection, meaning not because of normal microbiota. There is more normal microbiota, at least bacterial cells, on a person than there are human cells. There are some recent places where we found normal microbiota, and it came as a little bit of a shock to the microbiological community because just people didn't think that there was bacteria growing there. One would be inside the female bladder, we can find some normal microbiota. Another would be inside mother's milk, we can find normal microbiota. And the third, I'm not sure why that was a surprise, and that is the placenta can have a unique microbiota, meaning we can find cells there that we do not find anywhere else on a human normal microbiota. And why it's unusual is because the placenta only lasts during the pregnancy. And then after the pregnancy, the placenta is expelled. And so the normal microbiota will no longer continue, at least the placental normal microbiota. You should know the normal microbiota can inhibit the ability of a human pathogen to get established and then to grow and cause infection. We call this microbial antagonism, where two species of microbes are competing with each other. They can be competing for space. If there's not space there, then the human pathogen can't fall there and then get established and grow because there's no space for the normal microbiota. The normal microbiota can compete for resources. if they remove all the food and then Staphylococcus aureus were to fall on that area of your skin, there's no food around, so Staphylococcus aureus cannot grow. The normal microbiota can change the environment, making it less suitable for a human pathogen to get established. And the main way that normal microbiota do this are to grow in the media, secrete acids, and then the acid being produced in the media of the normal microbiota can be used to prevent the human pathogen from getting established, such as when Staphylococcus epidermidis on our skin digests the lipids, the skin wax, whatever you call that, skin oil, and then makes it more acidic. That will prevent many human pathogens from growing simply because it's too acidic. And then the final way that bacteria can engage in microbial antagonism with a normal, not a normally, a human pathogen is the bacteria can produce a bacteriocin. You can think of a bacteriocin being a very narrow, more narrow than any other bacteria, antibiotic, more narrow than any antibiotic, and only infects cells similar to the cell which secreted the bacteriocin. And so you can think of it as a very specific antibiotic. Any question about any of that? You should know the normal microbiota establishes a relationship with the host, which is either mutualistic symbiosis or commensalistic symbiosis, where the normal microbiota tends to benefit because it's given a home. Usually they're given food. And so the bacteria can go there and then grow. If the bacteria benefits the host, we call that mutualism. If the bacteria does not benefit the host and just does not damage the host, we call that commensalism. Example of commensalism was a small microscopic worm that each of us have one in our eye. And it's a nematode, and I'm guessing it's growing on the bacteria that fall into your eye, because the nematode does no damage to the human eye. Does no damage, but does not benefit the humans. So there's a commensalistic relationship. A parasitic symbiotic relationship is where a microorganism or microbe grows on the host and then damages the host, causing disease or signs and damaging the host. The host loses, and so we call this parasitism or parasitic symbiosis, where the two are living together, but one is damaged and the other benefits. The parasite is getting a host and food from the host. We might be killing the cell and then releasing the food. All right. The normal microbiota does not normally cause disease. It can under specific circumstances. When it does, we call it an opportunistic microbe, where a microbe is growing where it doesn't normally grow, and so we call it opportunistic. In a healthy individual, the normal microbiota does not normally cause disease, but in an immunocompromised patient, the normal microbiota could cause disease, such as with E. coli growing where it should not, like growing in the ear or in the lungs, causing disease. In the ear, it would be causing an ear infection or an earache, and in the lungs, it would be causing pneumonia. You should know about Cox postulates. There will be a question on Cox postulates on the final. You should know about the terms. You should know about the terms of disease, incidence, new cases only, prevalence, both new cases and old cases. So the prevalence will always be bigger than the incidence. In 2007, I'm giving you an example of AIDS, the incidence being 56,300 new cases in 2007. The prevalence is much larger, over 1 million cases of AIDS in the United States. Did you know about the occurrence of disease, severity of disease? A primary infection is where the acute infection causes the initial disease. It may weaken the host, allowing a secondary infection where another microbe is able to grow because the primary infection weakened the host, such as when somebody gets a sore throat from a viral infection, and then that weakens the patient's mucosa, and then they can get a bacterial infection in the throat. Subclinical disease is also an asymptomatic disease where the patient has no noticeable signs or symptoms. That doesn't mean they can't spread the disease, but they have no signs or symptoms of disease. You should know about herd immunity. The whole point of vaccinating the population is to create herd immunity, where if enough people are vaccinated, then you protect most of the people in the population that are not vaccinated. So red is a case where the patient has become infected with the organism causing disease. And if we have enough of the population immune, only people in very close contact or close proximity to the infected patient can get the disease. Another patient cannot get the disease, like this patient here, because the disease would have to go through either this patient or that patient to get to the patient. And these people are immune. So it's causing herd immunity, protecting all of these people over here. They're still blue. Any question about any of that? The example is if you have over 90% of children in the United States immune to measles, we have herd immunity. And then the few that are not immune will generally be protected, unless they're in close contact with somebody who gets the disease. The trouble is parents tend to not get their children vaccinated, and then the percentage of children who have immunity drop below 90%, and then suddenly the measles can then spread again. You should know about the patterns of disease. Disease has to come from a reservoir. You should know about the predisposing factors of the disease. And then you should know about the development of disease going through different stages of disease. First stage is incubation period, when the patient first gets infected, until the patient has signs or symptoms. That's the incubation period. Once the patient first has signs or symptoms, they're generally mild. Like when you first start feeling you have a sore throat and you say, geez, do I have a sore throat? That's the prodromal period. It ends when the signs and symptoms are no longer mild. That's the period of illness, when the signs and symptoms are no longer mild, till when the period starts getting better. That will begin the period of decline. an example of period decline and the period of illness, what breaks it would be when the child has a fever in the period of illness. When the fever breaks, we've now entered the period of decline. After the period of decline, we have the period of convalescence. In the period of decline, the number of organisms causing disease drops and drops. Once it drops below a certain threshold, we no longer have enough organisms to cause any signs or symptoms, and so the disease goes asymptomatic. And then the period of convalescence will continue from when the disease becomes asymptomatic till when the patient finally throws off the disease-carrying microbe. That's the period of convalescence. With the human cold, what I like to do when I first get it is I notice I'm coming down with a cold in the prodromal period. I then try to get lots of rest, and then I can skip the prodromal period, going to the period of illness and the period of decline, and I can move it into the period of convalescence. That only works with some colds. It does not work with the flu. I've tried. And this is stating that not all diseases have all stages of disease. Like when I successfully moved the human cold from the prodromal period to the period of convalescence, there is no period of illness or period of decline. And then if your patient gets HIV in the period of illness, and then the patient dies of AIDS, this will be another disease that does not go through all the stages of disease. Meaning in the AIDS patient that died, there is no period of decline, no period of convalescence. Any question about any of that? All right. You should know about the reservoirs of infection. There are 3 different. You should know about the transmission of disease. It happens in 3 ways: contact transmission, vehicle transmission, and vector transmission. Vehicle transmission is an object brings the disease-carrying organism to the healthy individual susceptible patient. And then vector transmission, a bite or a scratch or something of an animal brings the infectious agent to the healthy individual. Direct contact transmission is when the patient has direct contact with another patient, such as handshake. There's 3 different contact transmission: direct, indirect. Indirect would be when a patient touches a baseball, the baseball then spreads disease to other players on the team. So the baseball is a fomite spreading the disease. And then usually the patient will pick it up on their finger and then inoculate it into the mucous membrane of their face. And that's indirect contact transmission. There's also droplet transmission, which I won't test you on. And that's when the person coughs or sneezes, and within 1 meter, that's droplet transmission. Beyond 1 meter, it is vehicle transmission. And we're not going to talk about it, airborne transmission, because it's a little bit arbitrary. Oops. Where we draw the line there. And it doesn't really matter because it's the same cough or sneeze. Three types of vehicle transmission: waterborne, foodborne, and then airborne transmission. Vector transmission is where an animal carries the disease-carrying organism to the patient. And it can be mechanical, passive transport from the feet, or biological transmission, where the pathogen actually reproduces inside the animal. A case of biological would be malaria. Know about nosocomial infections. It's probably the most important topic we can teach you, and that is nosocomial infections is an infection a patient gets while visiting a health care facility, such as a hospital. They do not have the nosocomial infection until they check into the healthcare facility. They get the nosocomial infection in the healthcare facility, and between 5 and 15% of patients come down with a nosocomial infection. This causes a number of deaths, as well as a number of people getting infected each year from a nosocomial infection. As a healthcare worker, you should strive to prevent nosocomial infections. They come about because microorganisms are present in the healthcare environment, because the patient is compromised, and then there's a chain of transmission in the hospital, either direct contact transmission, indirect contact transmission, or vehicle transmission. Any question about any of that? All right. There are certain procedures you can follow to prevent spreading nosocomial infection. Always practice aseptic technique, proper handling of contaminating materials. You must have frequent and thorough hand washing. After you touch a patient's skin, you should always wash your hands before going to the next patient. Or if you have gloves on, you should take off the gloves, throw them away, and put on fresh gloves. Education of staff, patients, and visitors has actually gotten quite good in the United States, and we've decreased the number of nosocomial infectious agents by at least half in the last, I don't know, something like 10 or 15 years. All right, you should know about epidemiology, and that there are certain diseases that a physician or clinician are required to report by law. Usually they report it to the local county health authorities, who then take that information and report it to the state health authority, that then sends the information to the CDC in the United States, the Center for Disease Control, and then they analyze all United States epidemiological information and then publish it weekly in the Morbidity and Mortality Weekly Report. Any question about any of that? All right. Let's briefly go through chapter 8. You should know the terms of chapter 8. You should be able to describe DNA replication, RNA transcription, and protein translation. You should also understand the different types of mutation. You should know the terms. You should know about DNA. You should know about the vertical flow of DNA within a cell. This is known as the central dogma in biology. About the flow of information in the cell can be within a cell. It can also be between generations of cells, like a parent cell to two daughter cells, or when we're talking about humans, from two parents to one offspring. And then the flow of information can also move from one donor cell into one recombinant cell. And this is the flow of information between cells of the same generation. You should know that information, but I've already talked about it. And I'm not going to go into it in great detail. You should know about the operon. An operon is a super gene. Normally, when we're talking about a gene, we are talking about a structural gene. In the lac operon, there are 3 structural genes and 1 operon called the lac operon. The operon involves the terminator at the end of the gene, end of the structural gene, as well as the regulatory regions around the gene that control the expression of the structural gene. And that would be the promoter and the operator in the lac operon. You should know how the lac operon works. Why do I have that there? You should know about mutations. You should know about the frequency of mutation. When a cell is just replicating the DNA, about 1 in 10 to the 9th replicated basis will be a spontaneous mutation. A mutagen can increase this mutation rate from about 10 to 1,000 times greater. Now, here's something. You should know that in horizontal gene transfer can happen by one of 3 ways: by transformation, conjugation, or transduction. All right, that's it for chapter 8. Any questions about chapter 8? If not, let's go on to chapter 6. You should know the terms of chapter 6. You should understand the various requirements of cells for growth, understand the 4 phases of growth, and compare the different methods for measuring microbial growth. You know the chemical requirements of oxygen, and that oxygen is, in a sense, a toxin. Oxygen O2 itself is not toxic, unless to us. It is toxic to many anaerobes, but to aerobes it is not. But there are metabolites of oxygen which are toxic. There's singlet oxygen, superoxide free radicals, peroxide anions, and then hydroxyl radicals. All of these are toxic oxygen metabolites. And the cell has to have some way of neutralizing these toxic forms of oxygen. Otherwise, it would be toxic to even an aerobe cell. And it's actually these 4 toxic forms of oxygen that kill anaerobes. You should know the different types of media. You should know about the main ways that bacteria grow. They have 3 ways of growing, and that's by binary fission. Most bacteria grow by binary fission. A few bacteria grow by budding, and then a few bacteria produce reproductive spores, where one cell creates more than 1 reproductive spore, something like usually around 4. And so that would be 1 cell ends up with 5 cells because the parent cell doesn't die. And the 4 reproductive spores will mean 1 cell makes 5 cells. That's reproduction. And how that differs from an endospore, where 1 parent cell makes 1 endospore, the parent cell dies, and so 1 cell makes 1 cell. That is not reproduction. An endospore is a survival strategy. All right, any questions about chapter 6? Move on to chapter 5. Know the terms. I don't think I've got the right one here. I don't have my preliminary slide there. Understand what feedback inhibition is. Understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. We went into great detail talking about aerobic respiration, how it's the most efficient way for a cell to make ATP, and how it makes ATP. So you should know that. You should also know how aerobic respiration differs from anaerobic respiration. The two are generally the same. The difference is that the electrons in anaerobic respiration do not flow to oxygen. They flow to some other acceptor, final electron acceptor, which is not oxygen. I should know about protein denaturation, inactivating enzymes. Should know about ribozymes. You should know that, let's see if I can show that better. There we go. In a eukaryote, the electron transport chain is happening inside the mitochondria, as is aerobic respiration. It'll start in the cytoplasm with glycolysis, and then we'll move to the mitochondria to engage in the Krebs cycle, which actually happens inside the matrix of the mitochondria. And then the electron transport chain happens across the inner mitochondria membrane. So between the inner and the outer membrane is where the electron transport chain is happening. In a prokaryote, that's happening across the cell membrane. And then the hydrogen ions are actually pumped into the cell wall. You should know that fermentation is essentially glycolysis with one or two fermentation steps. The cell can use this to make ATP as well as aerobic respiration or anaerobic respiration. The fermentation, you only get the generation of 2 net ATP from 1 molecule of glucose, and that production of ATP always happens in glycolysis. There's 1 or 2 fermentation steps afterwards. Well, we should mention briefly about the interrelationship of carbohydrate, lipid, and protein catabolism. There are amphibolic pathways that can happen both ways, and they integrate metabolism, even in human cells and actually all animal cells, which are lousy at integrating metabolism. We can make, well, generally fats from either carbohydrates or protein. I think everybody knows that if you eat a high protein diet and simply eat too much protein, you're going to store that down as fat. The same thing with high carbohydrate diet. You will eventually store that down as fats. And I think everybody knows that. And that's because of the integration of metabolism. Plants are much better at making molecules and much better at integrating metabolism. And so they are much better at reversing chemical reactions. And if you don't know, a plant can make all of the molecules it needs from simply CO2 and water. Now, usually it'll make CO2 and water into glucose and then make the other molecule from glucose. Animals, as you know, cannot make glucose from CO2 and water. And then you should know about photosynthesis. There are two photosynthetic reactions. Where do I have that? There's non-cyclic photosynthesis, which generates glucose. And then there's cyclic photosynthesis, which only generates ATP. If the cell only needs ATP, it'll engage in cyclic photosynthesis. Obviously, it has to be a photosynthetic cell. And if the cell needs glucose, it'll engage in non-cyclic photosynthesis. All right, any question about any of that? I should mention that the electron donor can vary from sun. The electron will start from the chloroplast, sun hitting the electron of the chloroplast, and then the electron flowing from the chloroplast, but the energy is from sun. For animals, the electron flows from glucose or some other food that we're digesting. So the electron donor can vary. The electron carriers are generally NAD and FAD in animals, but actually in plants can be something similar, NADP plus. And then the final electron acceptor can vary. For aerobic respiration, it'll be oxygen. For anaerobic respiration, it'll be some molecule other than oxygen. For fermentation, it'll be an organic compound like pyruvate. And then for photosynthesis, it'll actually be the chloroplast will be the final electron acceptor, at least in cyclic photosynthesis. Any question about any of that? All right, let's move on to chapter 4. In chapter 4, you should know the functional anatomy of prokaryotic cells. I don't think we're going to talk about eukaryotic cells, or we won't have a question about that, with the exception of the what is that theory called? The endosymbiotic theory. So you should know how prokaryotes and eukaryotes differ. You should know the terms of this chapter. You should know how gram-positive and gram-negative cell walls differ. You should know the importance of taxis in bacteria, what taxis is and the importance of it. And then the last thing you should know, well, there's a few things, I guess. You should know about inclusions, where something is being accumulated in the cell at such high levels that the material starts precipitating from solution, causing the formation of an inclusion. You should also know what an endospore is and how it is a survival strategy. And then lastly, you should know about the endosymbiotic theory and how it explains how mitochondria and chloroplasts came to be in a eukaryote cell. Any questions about any of that? All right, let's quickly go through chapter 3. You should know the terms. You should know the sizes of organisms and the properties of microscopes. You should understand the difference between a light microscope and an electron microscope. You should understand why staining improves resolution, why it helps us see an object. You should know the three types of stains: a simple stain, a differential stain, and a special stain. And you should understand the gram stain, its procedure, and the basis of its differential staining. You should know the metric system because this map is showing you the 3 countries in the world that do not use the metric system. And of these 3 countries, 2 of them are actually switching over to using the metric system. Although the United States have been switching for a long time, so we're very slowly switching. And then the other reason why you should know the metric system is science and medicine use the metric system. You should know the parts of the microscope, understand about refraction, reflection. I already mentioned that. Just going through it quickly to see if there's something I want to talk about. You should know the procedure of the gram stain. All right. Any question about chapter 3? On chapter 2, I don't think there are any questions directly about chapter 2. However, I do expect you to understand the concepts of chapter 2. So when I'm talking about a molecule, I expect you to understand the properties and the concepts of the molecule. Okay? Any question about chapter 2? So I don't ask anything specifically about chapter 2, but I do assume that you know and understand the topic of chapter 2. And then chapter 1, I've already mentioned. You should know the history of microbiology, such as the major historical events in microbiology, who discovered them, and generally you don't need to know the year. Oh, and chapter 1, remember, is also the classification of microorganisms, so a little bit of chapter 10. Know the terms. You should know the importance of microbes to people and the ecosystem. And I think that's it. I mean, I could go into great detail about the history of microbiology, but if you know the important events and who discovered them, that will be enough. Let me go through briefly. Oh, you should know about biological naming started by Carolus Linnaeus. We still use his naming convention. The scientific nomenclature, also called the binomial nomenclature, because there's 2 names to each species, a genus name and a specific epithet. We always use 2 names to name a species and their Latin names. Wherever you go in the world, we use that Latin name as the species name. Whatever language you use, Homo sapien is the scientific name for humans. You should know that names are either italicized or underlined. The genus name is always capitalized. The specific epithet name is always lowercase. The name is Latinized, used worldwide. The name can either be descriptive or honor a scientist. You should know about the 3-domain system of life and how we classify organisms. You should know about the book calls it a cell type. It can also be called a super kingdom, and that is bacteria and archaea have cell types prokaryotes. Eukarya have cell types eukaryotes. You should know about the different kingdoms and the different domains. Remember, we're only going to look at the six kingdom approach. You should know about a little bit about Antony van Leeuwenhoek. He's the first person who started microbiology looking at cells, and he found microbes almost anywhere he looked, including, here's a picture of his drawing, he saw the very first bacteria. And bacteria were not seen again for another 100 years, or about a century, and that's simply because the microscopes were not good enough to see bacteria, but Antony van Leeuwenhoek's microscopes were good enough to see bacteria. Did you know about Louis Pasteur and how he disproved the theory of biogenesis, not theory of the theory of spontaneous generation? Here's the history of microbiology. Oh, and then you should know about the concept, only the term, emerging infectious disease. Any question about chapter 1? If not, that's it. Any questions on anything? Sorry this was so long, but that's the review for the final. If there's no questions, I'll wish you good luck and good fortune. And I'll see you maybe never again. All right. Goodbye.
Thank you.
You're welcome.